How do lymphoma biopsies diagnose cancer? Imagine feeling a swollen lymph node and wondering what's going on inside. That's where a biopsy comes in. It's a key step in figuring out if cancer is present and what kind it might be. When doctors suspect lymphoma, they often need to look at the tissue directly. They do this by removing a small sample from the lymph node or nearby tissue. This sample is then sent to a lab where specialists examine it under a microscope. The goal is to find abnormal lymphocytes, which are immune cells that have turned cancerous. There are several ways to get this tissue sample. The most common method is an excisional biopsy. In this procedure, a surgeon removes the entire lymph node or a large part of it through a small cut. This gives the pathologist plenty of tissue to analyze in detail. It's usually done under local anesthesia if the node is near the surface, or with sedation or general anesthesia if the node is deep inside the body. Sometimes, if removing the whole node isn't possible, doctors perform an incisional biopsy, taking just a part of the node. Another approach is a core needle biopsy. Here, a hollow needle is inserted into the lymph node, often guided by imaging tools like ultrasound or CT scans. The needle extracts a cylindrical piece of tissue, which is less invasive than surgery but still provides enough material for diagnosis. For very small or hard-to-reach nodes, a fine needle aspiration might be used. This technique uses a very thin needle to draw out cells, but it usually doesn't preserve the tissue structure well enough for detailed lymphoma classification. Therefore, it's less common for initial diagnosis but can be helpful for monitoring or other assessments. Once the tissue is collected, a specialized pathologist examines it closely. They look for abnormal lymphocytes and perform additional tests. Immunophenotyping helps identify specific proteins on the cell surface, which helps classify the lymphoma subtype. Molecular and genetic tests can detect unique markers or genetic changes associated with different types of lymphoma. These details are crucial because they influence treatment choices and prognosis. The tissue's structure and cell arrangement, studied through histopathology, help distinguish lymphoma from other diseases and confirm the diagnosis. The results from the biopsy tell us if lymphoma is present and whether it is Hodgkin or non-Hodgkin lymphoma. They also specify the subtype of non-Hodgkin lymphoma, which is important because there are over 70 different types. Each type may requiring a different treatment approach. This detailed diagnosis guides doctors in planning personalized treatment strategies and helps assess how advanced the disease might be. In summary, lymphoma biopsies diagnose cancer by providing a tissue sample that allows expert pathologists to identify cancerous lymphocytes, classify the lymphoma subtype, and help plan the best treatment. This procedure is a cornerstone of lymphoma diagnosis and is supported by additional tests and imaging to understand the extent of the disease. Always remember, if you or someone you know is undergoing testing, consulting healthcare professionals is essential for personalized care and accurate diagnosis.